Hello and welcome to this film for Carlam Cymru A2 revision in biology. My name is Mrs. Kavanaugh, and in this session we are going to look at respiration. Respiration is found within Unit 3 of A2 biology. The unit is titled Energy, Homeostasis, and the Environment. The assessment for this unit is a written exam of 2 hours. This constitutes 25% of the qualification as a whole. Let's look at the objectives and outcomes for this session. We will look at A. The reasons why we need respiration to take place, and B. Why we need respiration to release energy for cell metabolism. There are 4 films within the series for A2 biology. This film on respiration, a second film in this unit on anaerobic respiration and respiratory substrates. The other 2 films in the A2 schedule are from Unit 4. These have been provided by Mr. Davies from e-sgol gland fluid, and are a film on Section 4.1 on human reproduction and 4.2 on plant reproduction. Now let's turn to the specification and the specific reference for this film. As I mentioned earlier, this session is from Energy, Homeostasis, and the Environment, and this is Part 3. Respiration releases chemical energy in biological processes. This is further broken down into Part A, which is the need for all living organisms to carry out respiration to provide energy in the cell. In Part B, glycolysis as a source of triose phosphate, pyruvate, ATP, and reduced NAD, and resulting in the formation of acetyl coenzyme A, but the names of intermediates are not required. In Part C, the Krebs cycle as a means of liberating energy to produce ATP and reduced NAD with release of carbon dioxide. And finally, Part D, the role of reduced NAD and FAD as a source of electrons and protons for the electron transport system. Now we'll move on to look at our keywords. You should think of ways that you can actively remember the meanings of these keywords. Using mnemonics is a way of doing this and can help you to remember more effectively and efficiently. But not only knowing the meaning, but also knowing how to use the words in exam responses will help you to gain invaluable marks. We will look at some exam questions at the end of the session. The first word is hydrolysis. This is a common form of a chemical reaction where water is mostly used to break down the chemical bonds that exists between a particular substance. Hydrolysis is derived from Greek, hydro meaning water, and lysis, which translates to the word break or to unbind. Next, we have the inner mitochondrial membrane. This separates the matrix from the intermembrane space. The electron transfer chain is located here. The intermembrane space is the small space between the inner and outer mitochondrial membranes. Protons flow out of the intermembrane space across the inner membrane. The matrix is a fluid-filled space within the inner membrane of the mitochondria, which contains mitochondrial DNA and the enzymes needed for aerobic respiration. Protons flow across the inner membrane into the matrix. The mitochondrion is an organelle which is the site of aerobic respiration. Moving on to ATP or adenosine triphosphate. This is the energy storage and carrier molecule found in all living cells. ATP synthase is the enzyme found in cell membranes to phosphorylate ADP to form ATP as protons flow through it. Cytosol is the fluid part of the cell within the cytoplasm. The ETC or electron transport chain. Here, electron carrier proteins transfer electrons through oxidation-reduction reactions. Electron carrier, this is a molecule that can accept and donate electrons. A proton pump, which is an integral membrane protein which enables a proton gradient for ATP synthesis and phosphorylation. In phosphorylation, a phosphate is added to a molecule. There are things that we need to remember from our GCC work. We should remember some basics from respiration, including the site of aerobic respiration is the mitochondria. That aerobic respiration takes place with oxygen, and anaerobic respiration takes place without oxygen. And that ATP is the unit of energy currency. Now let's look at the reasons why we need to respire. There are a number of reasons why we need to respire. We need to carry out respiration in order to release the energy in our cells. We use the energy that has been released to enable our muscles to contract so that we can move and to enable cell division to take place. Let's have a look at a brief summary of aerobic respiration. We need to remember that respiration is a series of enzyme-catalyzed reactions. They are catabolic reactions. This means that larger complex molecules are broken down into smaller, simpler molecules. Aerobic respiration takes place in cells, and depending on the stage of aerobic respiration in the cytoplasm or the mitochondria. In aerobic respiration, we use glucose as our respiratory substrate. There are other substrates, but we will discuss these in our other film, which is energy-rich, and is broken down to give us energy. During aerobic respiration, some energy is transferred to make ATP, adenosine triphosphate, from ADP, adenosine diphosphate, and some energy is released as thermal energy. The structure of ATP is shown here. There are 3 parts to ATP. A ribose sugar joined to a base, which is adenine, and also 3 phosphate groups. You may be required to draw a diagram of ATP or to label a pre-prepared diagram. As I mentioned earlier, aerobic respiration is catalyzed by enzymes. Let's look at some of these enzymes. ATP hydrolase is used to hydrolyze ATP into ADP and an inorganic phosphate. This means that the phosphate produced can phosphorylate other compounds, making them more reactive. ATP synthase catalyzes the condensation reaction between ADP and the inorganic phosphate making ATP. These 2 diagrams show the places where aerobic respiration takes place. The mitochondria on the left shows some of the sites of aerobic respiration, and we will look at these as we move through the presentation. The diagram on the right shows a plant cell. Aerobic respiration takes place in plant and animal cells. In this film, we see someone running. They're using the process of aerobic respiration. This shows how the muscle contractions are working, using energy to enable the person to move. There are 4 stages in aerobic respiration, which we are now going to look at in detail. Stage 1 is glycolysis. Stage 2 is the link reaction. Stage 3 is the Krebs cycle. And stage 4 is oxidative phosphorylation. This diagram shows us the stages in aerobic respiration. Briefly, glucose, our respiratory substrate, enters the reaction. The process of glycolysis takes place. Pyruvate is produced and enters the link reaction. From the link reaction, acetyl-CoA enters the Krebs cycle. In the final stage, in the electron transport chain, oxygen is the final electron acceptor. This diagram shows us glycolysis. Glucose enters the pathway. Glucose is phosphorylated using the phosphate groups from 2 molecules of ATP. ATP is hydrolyzed into ADP and inorganic phosphate, thus providing the phosphates for glucose phosphorylation. This forms an unstable molecule, which breaks down immediately into 2 3-carbon molecules called triose phosphate or TP. Hydrogen is removed from TP to convert it to pyruvate. Hydrogen is transferred to a coenzyme called ND to form NADH or reduced NAD. To recap, glucose is phosphorylated using the phosphate groups from two molecules of ATP. ATP is hydrolyzed into ADP and inorganic phosphate, thus providing the phosphates for glucose phosphorylation. This forms an unstable molecule, hexose biphosphate, which breaks down immediately into two 3-carbon molecules called triose phosphate or TP. Hydrogen is removed from TP to convert it to pyruvate. Hydrogen is transferred to a coenzyme called ND to form an ADH or reduced NAD. Removing hydrogen from TP oxidizes it. The reduced ND is now used in the final stage of aerobic respiration in the electron transport chain during oxidative phosphorylation. This takes place in the cytosol of the cell. Glycolysis takes place in the cytosol. The conversion of TP to pyruvate produced 4 molecules of ATP. 2 molecules of ATP were used for phosphorylation of glucose in the first step. There is, therefore, a net gain of 2 ATP molecules in this stage of glycolysis. The pyruvate made at the end of glycolysis moves into the mitochondria for the next stage of aerobic respiration, which is termed the link reaction.
The link reaction takes place in the mitochondrial matrix. It joins the two processes of glycolysis and the Krebs cycle together. Pyruvate from glycolysis is converted into acetyl coenzyme A, which is shortened to acetyl-CoA. At this stage of the link reaction, no energy in the form of ATP is produced, but reduced NAD and acetyl-CoA are produced. Acetyl-CoA will be used in the Krebs cycle, and the reduced NAD will be used in oxidative phosphorylation. A carbon atom is removed from the pyruvate, forming carbon dioxide. Pyruvate now becomes a 2-carbon molecule. It is called acetate. Hydrogen is also removed, and is picked up by the coenzyme NAD, to form reduced NAD. The acetate joins with coenzyme A, CoA, to form acetyl-CoA. The link reaction takes place twice for each glucose molecule. This diagram summarizes the link reaction nicely. Pyruvate from glycolysis is converted into acetate, then to acetyl coenzyme A, which is shortened to acetyl-CoA. At this stage of the link reaction, no energy in the form of ATP is produced, but reduced NAD and acetyl-CoA are produced. Acetyl-CoA will be used in the Krebs cycle, and the reduced NAD will be used in oxidative phosphorylation. A carbon atom is removed from the pyruvate, forming carbon dioxide.
Let's find out what you have learned so far. Are you ready for a quick quiz? Read each of the 3 questions, then pause each film and write down your answer. When you have done this, then restart the film to check if you were correct. Where does the link reaction take place? Our choices are the cytosol or the matrix of the mitochondria. And the correct answer is the matrix of the mitochondria. How many carbon atoms are found in pyruvate? Is it 2 or 3? The correct answer is 3. Your third and final question of the quiz. How many carbon atoms are found in the acetyl group CoA? The answer is 2. How many did you get right?
Now let's look at the third stage of aerobic respiration, the Krebs cycle. In brief, the acetyl-CoA enters the Krebs cycle. Here glucose is oxidized. Carbon dioxide, ATP, reduced NAD and reduced FAD are produced. The reduced NAD and FAD will be used later, during oxidative phosphorylation. Now let's look at the stages of the Krebs cycle. The acetyl-CoA enters the Krebs cycle from the link reaction. The acetyl-CoA reacts with a 4-carbon molecule called oxaloacetate. The CoA part of acetyl-CoA is removed, to be reused. The oxaloacetate 4-carbon joins with the 2-carbon of acetyl to form a 6-carbon molecule called citrate. Carbon and hydrogen are removed. Carbon dioxide and reduced NAD are formed. Citrate is now a 5-carbon compound. Two processes now occur, decarboxylation and dehydrogenation. This converts the 5-carbon compound into a 4-carbon compound. ATP, 3 molecules of reduced NAD, 1 molecule of reduced FAD, and carbon dioxide, are also formed. This process takes place twice for each glucose molecule. Now, we'll look at the Krebs cycle process using this interactive diagram. At step 1, carbon dioxide has been removed. This enables acetyl to enter the Krebs cycle. At step 2, the oxaloacetate at step 3 joins with the acetyl group to form the citrate at step 4. Now carbon dioxide is removed. This is step 5. At step 6, hydrogen is removed via NAD to become reduced NAD. This leaves us with step 7, our intermediate molecule. We don't need to know the name of the intermediaries. This intermediate is a 5-carbon molecule. At step 8, carbon dioxide is removed and also hydrogen through NAD and becomes reduced NAD. A further intermediate molecule is formed at step 9. At step 10, hydrogen is lost through NAD becoming reduced NAD. We move on to step 11, our final 5-carbon molecule. Before finally, step 12, where hydrogen is removed again, and the 4-carbon oxaloacetate is ready to begin the Krebs cycle again. It would be a good idea to play this part of the film again, to familiarize yourself with the Krebs cycle. Now, we summarize the final stage of aerobic respiration. This is the part where most of the ATP is made. Here, the electrons carried by reduced NAD and reduced FAD from the first 3 stages of aerobic respiration, glycolysis, the link reaction and the Krebs cycle, are now used. This takes place across the inner mitochondrial membrane. There are 2 parts to the process, 1, chemiosmosis and 2, the electron transport chain. Now we'll look in more detail at the process of oxidative phosphorylation. The reduced NAD and reduced FAD from the previous stages release their hydrogen atoms. The electrons move to electron carriers, which are embedded within the inner mitochondrial membrane. Energy is lost as electrons move between the electron carriers. Hydrogen ions accumulate in the intermembrane space. The hydrogen ions then flow back into the matrix through the enzyme ATP synthase. At the end of the electron transport chain, the electrons are passed on to oxygen, the final electron acceptor. This diagram shows us what happens during this final stage of oxidative phosphorylation. The electron transport chain contains carrier proteins. Reduced NAD releases hydrogen, electrons transfer their energy to the proteins in the membrane, giving the energy for hydrogen ions to be pumped across the inner mitochondrial membrane. The flow of ions back across the membrane synthesizes ATP by the enzyme called ATP synthase. As oxygen is the final hydrogen ion and electron acceptor, it combines with the hydrogen ions and electrons to form water. This diagram of the mitochondrion highlights some of the main parts used in aerobic respiration. Intermembrane space, inner membrane, matrix, and cristae. The inner membrane is folded, forming cristae. This means that there is a greater amount of electron transport chain enzymes, and ATP synthase, in the mitochondrion.
It's exam question time. In this last section, you will find some practice exam questions. Take time to read each question carefully and try to work out what the examiner wants to know. Pause the film for each question, write down your answers and check if you are correct. If you are, then well done. If not, then go back to the relevant section in the lesson, to enhance your knowledge. Then try again. ATP is regarded as a universal energy currency, as it is used in all organisms, for cellular processes. Draw a simple, fully labeled diagram of ATP. Here is the diagram and the answers that were acceptable. Only one phosphate need be labeled. Pentagon shape with adenine and 3 phosphates coming off at correct points. 1. Ignore shape of adenine. Correct labels equals ribose plus adenine plus phosphate. Accept adenosine, if structures bracketed. Reject pentose. Reject base. Explain the importance of ATP in cells. Max 3. Here are the acceptable answers, check yours against them. References to a suitable function of ATP. E.g. protein synthesis, active transport, muscle contraction, not movement. Different types of energy can be transferred into a common form. Only one molecule needs to transfer energy to chemical reactions. Energy can be supplied, in small amounts, packages, approximately 30.6 kJ, less energy, heat, wasted. Easily transported, across membranes. Single enzyme, only ATPase, needed to release energy from ATP. Single bond needed to be broken, one-step reaction, to release energy. The detailed diagram opposite shows the link reaction in Krebs cycle. Most of the intermediates involved are named. Using the diagram, and your own knowledge, to determine how many carbons there are in the following Krebs cycle intermediates. Citrate. Alpha-ketoglutarate. Succinate. 2. Explain precisely how you have arrived at these figures. The answer for citrate is 6. The answer for alpha-ketoglutarate is 5. The answer for succinate is 4. The answer for part 2 is 4C oxaloacetate plus 2C acetyl. 1C lost, CO2 lost, before alpha-ketoglutarate, from isocitrate, isocitrate is decarboxylated. And 1C lost, CO2 lost, from alpha-ketoglutarate, before succinate, alpha-ketoglutarate is decarboxylated. Name the 2 enzyme types involved in the conversion of pyruvate to acetyl-CoA. Dehydrogenase, decarboxylase.
Thank you for watching this film. Revise well and prosper.